Identifying where regulatory proteins bind across the genome is essential for understanding the gene regulation. ChIP-seq provides a genome-wide approach to mapping these protein DNA interactions. ChIP-seq stands for chromatin immunoprecipitation followed by sequencing. It is used to identify genomic regions associated with a specific DNA binding proteins such as transcription factors as well as specific histone modifications. To understand how ChIP-seq works, we first need to consider the organization of the genome. DNA is packaged into chromatin, where it is associated with histone proteins and organized into dynamic structure. Regulatory proteins interact with this chromatin at specific genomic regions, influencing processes such as transcription and chromatin regulation. The ChIP-seq workflow begins by preserving protein DNA interactions, typically through cross-linking. The chromatin is then fragmented into smaller DNA fragments, commonly using sonication or enzymatic digestion. Next, an antibody specific to this protein or histone modification of interest is used to selectively isolate the associated chromatin fragments. This step is known as chromatin immunoprecipitation. The DNA is then purified from the isolated chromatin and prepared for high-throughput sequencing. The resulting sequencing reads are aligned to a reference genome regions, showing significant enrichment of reads relative to the background are identified through peak calling. A ChIP-seq peak therefore represents a genomic region with enriched signal, indicating that the protein or histone modification of interest is associated with that region. To distinguish true enrichment from background signal, ChIP-seq experiments commonly include an input DNA control, representing fragmented chromatin before immunoprecipitation. The biological interpretation of the peak depends on what is being studied. For transcription factors, peaks can identify candidate regulatory regions including promoters, enhancers, and regulatory elements. For histone modifications, their genomic distribution can provide information about chromatin states and regulatory activity. ChIP-seq can therefore be used to investigate transcriptional regulation, identify potential regulatory elements, study gene regulatory networks, and compare regulatory landscapes between different biological conditions, developmental stages, or disease states. However, ChIP-seq has important limitations. Its reliability depends on factors such as antibody specificity, experimental design, sequencing depth, background signal, and computational analysis. In addition, a ChIP-seq peak indicates enrichment at a genomic region, but it doesn't by itself establish that the interaction is functionally regulatory or causally affects gene expression. For this reason, ChIP-seq is often integrated with complementary approaches. RNA-seq can reveal changes in the gene expression, while AT-AC-seq can provide information about chromatin accessibility. Together with functional validation experiments, these approaches can help connect regulatory protein, beginning with chromatin states and transcriptional output. Ultimately, ChIP-seq transforms protein DNA interactions that cannot be directly observed at the genomic scale into measurable genome-wide map. By identifying where regulatory proteins and histone modifications are enriched, ChIP-seq provides a powerful framework for understanding how chromatin organization contributes to the regulation of gene expression.